Welcome to the deep dive. Today we're plunging headfirst into a topic that touches every single one of us, often without us even realizing it. The microscopic world of pathogens and infection.
It's everywhere, isn't it?
It really is. And let me kick things off with a truly sobering statistic. Infectious diseases currently cause about 1/4 of all human deaths worldwide.
Yeah, that's more than all forms of cancer combined. It's a staggering figure.
It really puts things into perspective.
Especially when you think about, ancient diseases like tuberculosis and malaria, they're still a huge burden, and then you've got drug resistance making it even harder.
Right, that rising resistance is scary.
And of course, we're constantly seeing new things emerge. COVID-19 is the obvious recent example, hitting hundreds of millions. It's a constant battle.
And it's not just the big pandemic threats either, is it? Sometimes the science completely flips what we thought we knew.
Oh, definitely. Like the stomach ulcer story.
Exactly. For years, everyone thought, oh, it's stress, it's spicy food.
Standard medical wisdom.
Turns out, most are actually caused by this tiny bacterium, Helicobacter pylori, just living in the stomach lining.
It completely changed the game for treating ulcers. A total paradigm shift, really, based on understanding the infectious cause.
So that's what we're doing today. This deep dive is all about exploring that fascinating, sometimes scary, world of pathogens and infection.
And our guide for this is a key chapter from Molecular Biology of the Cell, 7th edition. It really gets into the nitty-gritty of how these invaders work.
We're going to pull back the curtain, see how these microscopic invaders aren't just random attackers. They're actually master manipulators.
That's the key point. They exploit our own cellular machinery. They hijack our systems.
And by studying them, we learn so much, not just about disease, but about how our own cells work normally. It's kind of amazing.
It really is. So the mission today, for you listening.
Yeah, our goal is to give you a really comprehensive, clear, and hopefully engaging understanding. We'll cover the different kinds of pathogens, those intricate molecular dances between them and us,
how they evolve so darn quickly, and even the role of the microbes that live on and in us, our microbiota. Some of those are actually really helpful.
Right. Prepare for some valuable knowledge, maybe some surprising facts, definitely some aha moments. You'll leave feeling genuinely well-informed, ready to dive in.
Let's do it. So that global burden we mentioned, it's definitely not spread evenly.
No, you see huge disparities. Poorer countries, poorer communities often get hit much harder.
Right. Things like lack of clean water, sanitation issues, healthcare access, it all plays a huge role. The Haiti cholera outbreak in 2010 after the earthquake, a devastating example of that.
Such a tragedy.
But then you have other diseases like Legionnaire's disease that tend to pop up more in industrialized places, often linked to things like air conditioning systems.
So it's complex.
Yeah.
But fundamentally, these pathogens, they aren't acting out of malice, are they?
Not really, no. From their perspective, they're just organisms exploiting an available niche, trying to survive and reproduce like anything else.
And studying how they do that, how they exploit our cells, has actually been incredibly valuable for basic science.
Hugely valuable. They often target fundamental processes in our cells, so by figuring out what the pathogen is doing, we learn more about our own biology. It's a bit ironic.
And we should probably stress, most microbes out there are not harmful.
Oh, absolutely not. Only a tiny, tiny fraction are pathogens. Most of Earth's biomass is microbial. They're doing essential things like making oxygen, cycling nutrients in the soil.
And the ones living on us, our microbiota.
Many of those are beneficial, crucial even for our normal development, digestion, immune system function. They're partners mostly.
Okay, so we've seen the scale, the impact. Let's get specific. What exactly is a pathogen and what forms do they take?
Good question. We generally split them into two main types. You've got primary pathogens. These are the ones that can make healthy people sick.
Like SARS-CoV-2, the COVID virus, or Vibrio cholerae causing cholera?
Exactly. Then you have opportunistic pathogens. These usually only cause trouble if someone's immune system is weak, or maybe after surgery, something like that.
Like some strains of staph bacteria.
Precisely. And even among primary pathogens, there's variation. Some hit fast and hard, like cholera. Others can hang around for years, maybe without symptoms, like tuberculosis or certain parasitic worms.
Worms.
Right. So what are the main categories of these invaders?
Okay, first up, viruses. Tiny things. Basically just genetic material, DNA or RNA, wrapped in a protein coat, sometimes with an outer membrane.
The ultimate parasites, really. totally dependent on our cells.
Completely. They cause everything from COVID and Ebola to the common cold and polio.
Then there are bacteria, bigger, more complex cells.
Right, they're prokaryotes. They do most of their own metabolic stuff, but need us for nutrients. Think TB, pneumonia, lots of diarrheal diseases, STDs.
And beyond those two.
We get into eukaryotic organisms. These are more complex, like our own cells. This includes protozoa, single-celled eukaryotes.
Malaria is caused by one of those, right? Plasmodium. Yes, and African sleeping sickness caused by trypanosoma. Then there are fungi yeasts or molds, things like candida or histoplasma.
And finally,
the worms, the metazoa, multicellular parasitic animals, like asparagus, that gut nematode you mentioned. It can get up to 30 centimeters long inside a person.
that's unsettling. Okay, so viruses, bacteria, eukaryotes, no matter the type, they all need to follow a kind of playbook to succeed.
Pretty much. A 5-step plan, you could say. 1. Get in. Enter the host, usually breaching a barrier like skin or gut lining.
2. Find a home. Locate a suitable niche inside the host where they can get nutrients.
3. Hide or fight. Avoid or disable the host's immune system. This is crucial.
4. Multiply. Replicate using the host's resources.
And 5. Get out and spread. Exit the current host and find a new one to infect. That's the cycle.
That makes sense. Let's focus on bacteria for a bit. You said they're diverse. How do scientists even classify them?
Well, shape is one way rods, spheres called cocci, or spirals like the Lyme disease spirochete, Borrelia. But a really classic and still super useful method is gram staining. It tells you a lot about their cell surface structure.
Ah, yes, gram-positive and gram-negative. I remember that from biology class.
Exactly. Gram-positive bacteria, like strep or staph, have a thick outer cell wall made of peptidoglycan.
And gram-negative.
They have a thinner peptidoglycan layer, but it's sandwiched between two membranes, an inner and an outer one. That outer membrane has something called lipopolysaccharide, or LPS-E. coli, salmonella. Those are gram-negative.
And both that peptidoglycan and LPS are like red flags for our immune system.
Precisely. They're called PAMPs, pathogen-associated molecular patterns. Our innate immunity is primed to recognize them immediately. It's like a bacterial fingerprint.
What about things on their surface, like little swimmers or grabbers?
Yeah, they have appendages too. Flagella are whip-like tails for swimming. Vibrio cholerae uses those. And pili are hair-like structures, often used for sticking to surfaces, or even for exchanging DNA between bacteria.
Exchanging DNA. We'll need to come back to that. What about where they live? Do they all need a host?
Not necessarily. Some are facultative. They can live in the environment but cause disease if they get into a host. Others are obligate pathogens, meaning they have to be inside a host to replicate.
And do they stick to one type of host?
It varies. Some are generalists, infecting many species. Others are highly specific, like the salmonella strain that causes typhoid fever. It only infects humans. That's why Typhoid Mary was such a problem.
Okay, back to that DNA swapping. You said pili could be used for that? How does a generally harmless bacterium suddenly become dangerous? Is that related?
It often is. It's largely down to something called horizontal gene transfer. This isn't inheriting genes from a parent. It's getting genes sideways from other bacteria.
Like trading genetic upgrade card.
Kind of, yeah.
Yeah.
There are 3 main ways. Transformation is picking up bits of DNA floating around in the environment. Transduction is when bacterial viruses, called bacteriophages, accidentally carry DNA from one bacterium to another.
And the third?
Conjugation. That's a direct transfer, often via pili, where one bacterium passes DNA like a plasmid directly to another.
Wow, so they can just acquire new abilities.
Exactly. And genome sequencing has shown this is huge for bacterial evolution. Genes that make bacteria virulence factors are often clustered on these mobile bits of DNA, like pathogenicity islands or plasmids.
Yeah.
It lets them rapidly adapt to new niches, including us.
So a species isn't fixed. It has a core set of genes, but then this extra toolkit.
Precisely. We talk about the core genome that all strains share, and the much larger pangenome, which includes all the genes found across all strains of that species. It reflects their incredible adaptability.
Is there a good example of this in action, making a pathogen worse?
Vibrio cholerae is a perfect case study. The really dangerous pandemic strains evolved by picking up bacteriophages carrying the cholera toxin genes.
Ah, so they acquired the weapon.
Exactly. And they also acquired other pathogenicity islands. Plus, they can change their surface antigens the molecules our immune system recognizes. They switched from one type, O1, to another, O139, which helped them evade existing immunity and cause new waves of disease like that awful Haiti outbreak. It's a constant evolutionary arms race.
An arms race
Yeah.
fueled by gene swapping. Okay. How else do bacteria cause harm? Toxins seem like a big one.
Definitely. For bacteria that stay outside our cells, secreting toxins is a major strategy. These proteins diffuse away, find specific host cells, bind to them, and then mess things up inside.
Like tiny guided missiles.
Sort of. Many bacterial toxins have two parts. A B subunit that binds to the host cell receptor, providing specificity, and an A subunit that has the enzymatic or toxic activity that gets delivered inside.
You mentioned cholera toxin earlier. How does that work again?
Its A subunit gets inside intestinal cells and chemically modifies a key signaling protein called Gs. It locks Gs in the on state.
And what does it do?
It leads to continuous activation of another enzyme, adenyl cyclase, which makes tons of a messenger molecule called cyclic AMP or CMP.
Okay. And high CMP causes?
Massive secretion of chloride ions and water into the gut. That's the severe diarrhea, which grimly helps spread the bacteria to new hosts via contaminated water.
Wow. Are there other toxins like that?
Oh, yes. Anthrax toxin from Bacillus anthracis is another example. It actually has one binding B part, but two different A parts it can deliver.
Two different weapons.
Right. One, called lethal factor, is a protease that messes up signaling pathways leading to shock and death. The other, edema factor, is an adenyl cyclase itself, boosting CAMP and causing fluid buildup, or edema.
So secreting toxins are one weapon. What about bacteria that get up close and personal?
Some use contact-dependent secretion systems. They physically touch a host cell and inject proteins directly into it, like a molecular syringe.
You mentioned the type 3 system before, the injectisome.
Yeah, it looks remarkably like the base of a flagellum, suggesting they evolved from a common ancestor. It injects bacterial effector proteins right into the host cell cytoplasm. Pathogens like EPEC and Salmonella use this.
EPEC, the one that makes pedestals.
We'll get to that. And there's also the type 5e secretion system. This one's related to the machinery bacteria use for conjugation, that DNA swapping. H. pylori uses this, so does Legionella.
And the point of injecting these proteins is
to manipulate the host cell, block immune responses, trigger the cell to engulf the bacterium, create a safe place to live inside. All sorts of tricks.
Okay, let's shift gears slightly. What about the eukaryotic pathogens? Protozoa, fungi, worms. Are they playing a similar game?
Broadly, yes, but with added complexities. One big challenge for us is treating them. Because they're eukaryotes, like our cells, it's harder to find drugs that kill them without harming us. Less selective toxicity.
That makes sense. What about their life cycles?
Often very complex. Many pathogenic fungi show dimorphism.
Dimorphism. Two forms.
Exactly. They can switch between being a single-celled yeast and a multicellular filamentous mold. Often the switch is linked to infection. Histoplasma, for instance, is a mold in the soil, but switches to a yeast form when inhaled into the warm lungs, which is when it causes disease.
So the environment triggers the change?
Often, yes. Temperature is a common cue. Candida albicans, another common fungal pathogen, also does this.
And protozoa, like malaria.
Even more elaborate life cycles. Malaria is maybe the most devastating protozoal disease. Hundreds of millions infected. Its parasite, Plasmodium, needs both a human and a specific type of mosquito, Anopheles, to complete its life cycle.
Two hosts.
And it transforms into multiple different forms, each specialized for invading a specific place. Mosquito gut, human liver cells, human red blood cells. It's incredibly adapted.
Just amazing complexity. Okay, what about viruses? You called them the ultimate hitchhikers.
Because they are. They rely almost completely on the host cell's machinery. They basically just carry their genetic blueprint DNA or RNA and maybe a few key enzymes. But they need all ribosomes to make proteins or energy, often our enzymes to copy their genome. Everything.
Their genomes can be DNA or RNA.
Yep. Double-stranded DNA, single-stranded DNA, positive sense RNA, negative sense RNA, even RNA that gets reverse transcribed into DNA, like HIV, huge variety.
The basic life cycle is similar.
Generally, yes.
Okay.
6 steps. 1, get in the cell. 2, uncoat release the genome. 3, replicate the genome. 4, make viral proteins using host ribosomes. 5, assemble new virus particles. 6, get out.
And one virus particle going in can lead to thousands coming out.
Easily. They're incredibly efficient replicators.
What do they actually look like?
All sorts of shapes and sizes. From tiny parvoviruses to recently discovered giant viruses that are almost as big as bacteria, the core is the genetic material inside a protein coat, the capsid. Capsids are often highly symmetrical, like an icosahedron, think poliovirus, or a helix. Some viruses also steal a bit of host cell membrane as they exit, forming an outer envelope.
They steal our membrane.
Yes, often by budding from the plasma membrane. Doesn't necessarily kill the cell immediately. HIV does this.
Given how much they rely on our machinery, Is it hard to develop antiviral drugs?
Extremely hard. You need to find drugs that block the virus but don't harm our own cellular processes. That's why vaccination has been and remains our best weapon against many viral diseases.
Like smallpox eradication.
The perfect example. A global vaccination campaign wiped it out by 1980, and polio is nearly gone thanks to a similar effort since 1988. Vaccines are true public health triumphs.
Okay, we know who the players are. Now, how do they actually breach our defenses and get inside? It seems like our bodies should be pretty well protected.
They are, but pathogens have evolved ways around those defenses. The simplest entry is just a break in the skin, a wound.
Easy access.
Right. Pathogens like Staphylococcus, including nasty MRSA or papillomaviruses causing warts, can get in that way. No fancy mechanisms needed.
What about intact skin?
That's where arthropod vectors come in. Mosquitoes, ticks, fleas. They bypass the skin barrier with their bite. Very efficient delivery system.
And they can carry diseases from animals to humans, zoonoses.
Exactly. Malaria, Plasmodium, Zika virus, yellow fever virus, Lyme disease, Borrelia. Many rely on these vectors. The pathogen often has to replicate in both the insect and the mammal.
You mentioned Yersinia pestis, the plague bacterium manipulating the flea.
Yes. It grows in the flea's gut, blocking it. The flea keeps biting because it's starving, but it can't swallow, so it basically vomits bacteria into each new bite wound. Horribly effective for spreading.
Chillingly clever. What about pathogens that try to colonize surfaces like our airways or gut? They have defenses too, right? Mucus, cilia.
Absolutely. Mucus traps microbes. Cilia in the airways sweep the mucus up and out. Fluids flush things away. Pathogens need specific tools to overcome these.
Like sticky molecules.
Precisely. Adhesins. These are proteins on the pathogen surface that bind tightly to specific molecules on our epithelial cells. Uropathogenic E. coli, UPEC, causing UTIs, is a great example.
How so?
It has long pili sed that reach through the mucus layer. The tips of the pili have adhesins that bind specifically to certain sugars on bladder cells or kidney cells, determining where the infection takes hold.
And what about H. pylori in the superacidic stomach? How does it stick around?
It's got multiple tricks. Flagella for swimming through mucus to the less acidic zone near the cells. It produces urease, an enzyme that generates ammonia to locally neutralize the acid.
Creating its own little safe zone.
Exactly. And it uses that type IV secretion system to inject a protein called KGA into stomach cells. KGA messes with cell signaling, causes inflammation, and contributes to ulcers and even cancer risk over time.
Remarkable adaptation. So some stay outside cells but still cause trouble.
Yes, extracellular pathogens. Bordetella pertussis, the whooping cough bacterium, is like that. It sticks to airway cells using adhesins.
And then produces a toxin.
Right, pertussis toxin. Similar mechanism to cholera toxin, messes with the G protein, boosts CMP. This interferes with immune cells and causes that characteristic severe cough. The cough helps spread the bacteria.
Then there's that EPEC bacterium you mentioned, enteropathogenic E. coli, the one that makes pedestals.
Yes, EPEC. It causes diarrhea. It uses its type 3 injector system to shoot a protein called TIR into the host intestinal cell membrane.
It injects its own receptor.
Effectively, yes. TIR embeds in the host membrane and then acts as a docking site for another bacterial protein on the EPEC surface called intimin.
So the bacteria makes its own docking station on our cell.
Precisely. And when TIR binds intimin, it triggers this amazing response inside the host cell, massive actin polymerization. It builds a literal pedestal of actin filaments beneath the bacterium.
Pushing the bacterium up. What's the point of that?
It's not entirely clear, maybe helps with colonization or spread, but it definitely contributes to the diarrhea. What's really cool for scientists is that studying how EPEC triggers this has taught us a huge amount about how our cells normally control actin assembly, pathogens as research tools.
That is wild. Okay, let's talk about the ones that go all the way. Intracellular pathogens. Why live inside a cell?
Big advantages. You're safe from antibodies, harder for immune cells like phagocytes to get to, you've got a rich supply of nutrients, and access to the host's protein-making factory.
But you have to get in first. How do viruses do that? They bind to receptors. Then what?
Right. They co-opt host surface proteins as receptors. HIV needs CD4 plus a co-receptor like CCR5. People with a certain CCR5 mutation are actually resistant or less susceptible to HIV.
So binding is key. Then entry.
For enveloped viruses, membrane fusion is common. HIV fuses directly with the cell's outer membrane. Influenza gets taken inside via endocytosis first, then fuses its envelope with the endosome membrane, often triggered by the acidic pH inside the endosome.
What about viruses without an envelope?
Different tricks. Poliovirus forms a pore in the endosome membrane and squirts its RNA genome through. Adenovirus just disrupts the whole endosome membrane to escape.
Okay, viruses are small. Bacteria are much bigger. How do they get into cells that aren't normally phagocytic, like epithelial cells?
They often trick the cell into engulfing them two main ways: the zipper and trigger mechanisms.
Zipper.
The bacterium displays a protein, an invasion, that binds super tightly to a host cell adhesion molecule, like an integrin or E-cadherin. Think Yersinia or Listeria. This tight binding zips the host membrane around the bacterium.
And trigger?
This is more dramatic. Bacteria like Salmonella use their type 3 injector to pump effector proteins into the host cell. These proteins trigger massive rearrangements of the host cell surface, forming big ruffles that fold over and engulf the bacterium. It looks like macropinocytosis.
So one is a gentle zip, the other is a dramatic grab.
Kind of, yeah. Both hijack host cell machinery to get inside.
What about eukaryotic parasites, like toxoplasma or malaria?
They're often more active invaders. Toxoplasma uses a unique structure called a conoid and its own actin-myosin motor to literally burrow into the host cell.
It powers its own way in.
Yes. And as it enters, it forms this special vacuole around itself, the parasitophorous vacuole. It even removes host proteins from the vacuole membrane, making it resistant to fusion with lysosomes and selectively permeable to nutrients. Very sophisticated. Malaria parasites use a similar strategy.
Okay, once any of these pathogens get inside, they face the danger zone. The lysosome, the cell's recycling center, and garbage disposal full of digestive enzymes. How do they avoid that?
Critical step. Various strategies. Some just break out quickly. Listeria secretes a toxin, Listeria lysin O, that punches holes in the phagosome membrane, letting the bacteria escape into the nutrient-rich cytosol. Trypanosoma cruzi, causing Chagas disease, does something similar.
So escape into the cytoplasm. What if they stay in the vacuole?
Then they have to prevent it from becoming a lysosome. Mycobacterium tuberculosis is a master at this. It arrests the maturation of the phagosome so it never fully acidifies or fuses with lysosomes. It just hangs out in the stalled compartment.
Clever. Any other tricks?
Salmonella actually modifies its vacuole. It injects proteins using a second type 3 system once inside. These proteins recruit host motor proteins to pull membrane tubules out from the vacuole, creating this unique network called the Salmonella-containing vacuole, SCV.
Wow. And Legionella, the Legionnaire's disease one.
That one's really unusual. After being engulfed, it uses its type 4V system to inject effectors that prevent lysosome fusion and actually recruit bits of the host's endoplasmic reticulum membrane. It remodels the phagosome into an ER-like compartment where it can replicate.
Camouflage. It's amazing the diversity of strategies. Viruses manipulate membranes too, right?
Oh, yeah. Many enveloped viruses like SARS-CoV-2 induce the formation of complex membrane structures derived from the ER, creating protected replication factories. Then they acquire their final envelope by butting through other compartments like the ER-Golgi intermediate compartment. They really reorganize the cell's internal membranes.
Okay, so they're inside, they found a niche. How do they move around? The cytoplasm is crowded.
It is. Simple diffusion is too slow. So they hijack the host cytoskeleton. We talked about EPEC making pedestals outside the cell.
Right.
Well, inside the cell, bacteria like Listeria, Shigella, Rickettsia, and even some viruses like Ebola do something amazing. They induce actin polymerization at one end of themselves.
Building their own rocket engine.
Exactly, an actin comet tail that pushes them through the cytoplasm. Boom!
That's incredible.
And this movement isn't just random, it can propel them right into neighboring cells. They push out the membrane of the first cell into a long protrusion, which gets engulfed by the next cell.
So they can spread cell to cell without ever going outside.
Precisely. Avoids antibodies and other immune defenses in the extracellular space. And again, studying this bacterial actin rocketry taught us fundamental things about how our cells control actin dynamics.
What about the other cytoskeletal network, microtubules?
Viruses often use those, especially ones that travel long distances, like in neurons. Herpes viruses, like the chickenpox virus, hitch rides on motor proteins. Dynein motors move them towards the cell body for latency. Kinesin motors move them back out towards the axon tips for spreading.
Like riding cellular trains.
Pretty much, yeah. Along the microtubule tracks.
Our cells must have ways to fight back against these intracellular invaders, right?
They do. One key defense is autophagy, specifically a version called xenophagy, or antimicrobial autophagy. The cell basically wraps the invader in a membrane bag, an ontophagosome, and sends it to the lysosome for destruction.
But pathogens have countermoves.
Of course. Some disguise themselves to avoid being recognized by the autophagy machinery. Listeria uses its actin tails to literally outrun the forming autophagosome.
And some even turn it to their advantage.
Yes. Coxiella burnetii, Q fever, actually thrives in a vacuole that fuses with autophagosomes. It seems to use them as a source of nutrients. Poliovirus seems to use autophagy pathways to help it get trafficked and released from the cell. It's complex interplay.
It really is. Let's talk about viruses taking over the cell's basic operations. How do they ensure the cell makes viral stuff instead of its own?
They're masters of metabolic hijacking. Many encode proteins to shut down host gene expression. Poliovirus makes a protease that chops up a key component needed for transcribing most host genes.
Just turns off the host factory.
Largely, yes. Influenza blocks the proper processing and export of host messenger RNAs from the nucleus. They want the resources, ribosomes, nucleotides, energy, all for themselves.
And they mess with protein production, translation.
Definitely. Many inhibit the start of translation for host mRNAs. Some viruses, like influenza again, even steal the special cap structure from the front end of host mRNAs and stick it onto their own viral mRNAs. Cap snatching.
Stealing parts.
Or they bypass the need for a cap altogether, using special sequences in their own RNA called internal ribosome entry sites, or IRS, to grab ribosomes directly.
What about DNA viruses? Like adenovirus. They need to copy their DNA.
Right, and they often need the host cell's DNA polymerase, which is most abundant when the cell is actively preparing to divide in S phase.
So they force the cell to divide.
Essentially, yes. Adenovirus encodes proteins that disable key breaks on the cell cycle, like the RB and P53 proteins. This pushes the cell into S phase, ensuring plenty of DNA polymerase is available for the virus.
But messing with cell cycle breaks, doesn't that sound like cancer?
It can be, yes. That's why some DNA viruses are associated with an increased risk of certain cancers. They're removing the safeguards against uncontrolled cell growth.
And RNA viruses, how do they copy their RNA genomes? We don't usually have enzymes for that.
Most animal RNA viruses encode their own RNA-dependent RNA polymerases. They bring the tool they need. Retroviruses like HIV are special. They have an RNA genome but carry an enzyme called reverse transcriptase.
Which makes DNA from RNA.
Exactly. It makes a double-stranded DNA copy of the viral RNA genome, and then the DNA copy gets integrated, inserted right into the host cell's own chromosomes.
Permanently.
Pretty much, yeah. Becoming part of the host genome. That's latency for HIV.
This all leads back to that idea of an arms race. Pathogens evolve so incredibly fast.
Mind-bogglingly fast. Their generation times are tiny compared to ours, and the selective pressure from our immune system and drugs is intense. Think about it. Humans and chimps differ by about 2% genetically after millions of years. Poliovirus can change 2% in less than a week.
Wow, how does this rapid evolution manifest? How do they stay ahead?
One major strategy, especially for parasites and some bacteria, is antigenic variation, changing their surface coat to evade antibodies.
Like changing clothes constantly.
That's a good analogy. Trypanosoma brucei, the sleeping sickness parasite, is the classic example. Its surface is covered in one type of protein, VSG, but it has about 1,000 different genes for slightly different VSGs in its genome.
Well, it's disguises.
Basically, it uses gene rearrangement mechanisms to switch which VSG gene is active. So just when your immune system mounts a response against one VSG, a few parasites switch to a new one, escape, and start multiplying again. Causes those waves of sickness.
And bacteria do this too.
Oh, yes. Neisseria, the bacteria causing gonorrhea and meningitis, are masters at varying their pilin proteins, the ones used for attachment. They swap bits of DNA in and out of the active pilin gene from silent copies elsewhere in their genome.
Plus, they can pick up DNA from the environment.
Right. Neisseria are naturally competent for transformation. All this variability makes developing vaccines against them extremely difficult.
What about viruses? How do they vary?
Often through error-prone replication, their polymerases make mistakes, and they don't always fix them. HIV's reverse transcriptase is notoriously sloppy, no proofreading.
So it makes lots of mutations.
Loads. About 1 mutation per new genome, on average. This creates huge diversity within a single infected person. It's why drug resistance can pop up so fast, and why the virus can even change which cell types it prefers to infect over time. And it's a nightmare for vaccine design.
That rapid mutation sounds like a recipe for trouble. Is that how new flu strains emerge too?
Influenza has another trick up its sleeves: reassortment. Unlike HIV, its genome isn't one long strand. It's segmented, usually into 8 separate RNA pieces.
Like 8 mini chromosomes.
Sort of. Now imagine a cell, maybe in a pig, gets infected with 2 different flu strains, simultaneously, say, a human flu and a bird flu.
Okay.
When new virus particles are being assembled inside that cell, the RNA segments from both parent viruses can get mixed and matched, shuffled like a deck of cards.
Creating completely new combinations.
Exactly. Entirely new hybrid viruses. This antigenic shift, as it's called, is how major pandemic strains arise. The 1918 Spanish flu likely involved bird flu genes jumping to humans. The 2009 H1N1 swine flu had a mix of genes from human, bird, and pig viruses. It's unpredictable and can happen suddenly.
That constant evolution is why drug resistance is such a massive problem, isn't it?
It's the core of the issue. Antimicrobial drugs, especially antibiotics, have been miracle drugs. Penicillin in World War II saved countless lives. They work because they target things unique to bacteria-selective toxicity.
Like building their cell walls or specific bacterial enzymes.
Right. Things we don't have or that are different enough in our cells.
So what does this all mean for us? This rapid evolution of pathogens means drug resistance is a growing, serious problem. It feels like we're losing ground.
It's a huge concern. Resistance develops incredibly fast. New antibiotics often see resistance emerge within just a few years. With HIV drugs or malaria drugs, it can be months.
Months. That's terrifyingly quick. How do they actually become resistant? What are the mechanisms?
Generally, 3 main strategies. 1, change the target. Mutate the bacterial protein that the drug binds to, so the drug doesn't stick anymore.
Okay, modify the lock so the key doesn't fit.
Exactly. 2, destroy the drug. Produce an enzyme that chemically modifies or chops up the antibiotic, like beta-lactamases that chew up penicillin.
Fight back directly.
Yep. And 3, block access. Prevent the drug from getting to its target in the first place, maybe by reducing uptake, or more commonly, by pumping the drug back out of the cell using efflux pumps.
Pumping it out as fast as it comes in.
Pretty much. And the genes conferring these resistance mechanisms are often on those mobile genetic elements we talked about, plasmids, transposons.
So they can spread easily.
Very easily.
Yeah.
Through horizontal gene transfer, one bacterium becomes resistant, and it can pass those resistance genes to others, even different species. You get multi-drug resistant strains emerging. Think MRSA, vancomycin-resistant enterococci.
It seems like they have a ready-made toolkit for resistance. Where did these resistance genes even come from originally?
That's another fascinating piece. Almost all our antibiotics are natural products made by soil bacteria or fungi, like Streptomyces or Penicillium.
They make them naturally. Why?
As weapons to compete with other microbes in their environment. So in nature, microbes have been fighting each other with these compounds for millions of years.
Which means
Resistance mechanisms have also co-evolved over millions of years. If you sample soil bacteria that have never seen human-made antibiotics, you find they already have genes conferring resistance to many of our modern drugs.
So there's a vast environmental reservoir of resistance genes out there.
Exactly. And under the selective pressure of antibiotic use, pathogens can acquire these genes from environmental bacteria.
And our own behavior makes it worse.
Undeniably. Overusing antibiotics, using them for viral infections where they do nothing, using them extensively in agriculture, often just to promote animal growth, all of this creates intense selective pressure that favors the survival and spread of resistant microbes.
Which can then pass those resistance genes onto human pathogens.
That's the fear. We're accelerating the evolution of resistance through misuse.
Okay, pathogens are formidable. But you mentioned earlier, we're not alone. We have our own microbial communities, the microbiota.
Yes, our unseen partners. And here's a mind-blowing fact. You have roughly as many microbial cells in and on your body as you have human cells, maybe even slightly more.
Wait, really? More microbe cells than human cells?
By cell count, yes. Around 4 by 13 microbial cells versus about 3 by 13 human cells. You are, in a sense, a superorganism.
Wow. Where do they all live?
Mostly in specific places: skin, mouth, vagina, and especially the digestive tract. The large intestine is microbial headquarters, packed with thousands of different species.
And it's different for everyone.
Very different. Your microbiota is unique to you, influenced by genetics, how you were born, vaginal versus C-section, diet, age, hygiene, antibiotic use. It's a dynamic ecosystem.
What kind of relationship do we have with them? It's not all parasitic like pathogens.
Mostly not, no. It's usually commensalism they benefit, we're unaffected, or mutualism, we both benefit. Though some harmless commensals can become opportunistic pathogens if conditions change, like if the immune system weakens.
So they mostly help us, or at least don't harm us. How do they help?
In profound ways. Metabolically, for instance, their combined genes, the microbiome, contain maybe 100 times more genes than our own human genome.
100 times.
Yeah, this gives us huge extra biochemical capacity. Gut bacteria help digest food components we can't break down ourselves, producing beneficial molecules like short-chain fatty acids that influence our metabolism and even our immune system. They can even metabolize drugs we take.
So they're like a whole extra organ system, metabolically speaking.
In a way, yes. They're also crucial for development. Studies comparing regular mice with germ-free mice, raised completely sterile, show the microbiota is essential for the normal development of the gut lining, its structure, mucus layer, how quickly cells replace themselves.
And immunity.
Absolutely critical. The microbiota constantly educates our immune system, especially in the gut. It helps teach the immune system to tolerate beneficial microbes while remaining ready to attack dangerous pathogens. It influences the development of immune tissues and the balance of different immune cells. It's a constant dialogue.
So a healthy microbiota is important for a healthy immune system. What happens if it gets out of balance?
That's called dysbiosis, and it's being increasingly linked, correlated at least, with a whole range of diseases, autoimmune conditions, allergies, obesity, inflammatory bowel disease, maybe even diabetes.
Correlation isn't causation, but the links are suggestive.
Very suggestive. One really clear example of cause and effect is Clostridium difficile colitis. This nasty gut infection often happens after antibiotics wipe out the normal gut microbiota, allowing C. diff to overgrow.
And the treatment.
Remarkably, one of the most effective treatments is a fecal microbiota transplant. Transferring gut microbes from a healthy donor can restore the balance and cure the infection. It's powerful evidence for the importance of a healthy microbiota.
A literal ecosystem transplant. What's the next step in understanding all this?
The big challenge is moving beyond correlations to proving causation for many of these disease links. It's incredibly complex, thousands of microbial species interacting with each other and with us. But as we learn more about individual microbes and the molecules they produce, the hope is we'll gain huge insights into our own biology and find totally new ways to treat disease by manipulating the microbiota. It's a major frontier.
What an incredible journey we've taken. From the molecular tricks of viruses and bacteria trying to invade us to this vast, complex ecosystem living inside us that's actually vital for our health.
It really covers the spectrum, doesn't it? Understanding these interactions, pathogen host, microbe host, it's not just about fighting disease. It teaches us fundamental things about cell biology, evolution, immunity, life itself.
Every trick a pathogen uses, every way our microbiota helps us, it reveals something about the intricate workings of our own bodies.
Absolutely. Which leads to maybe a final thought to leave our listeners with.
Go for it.
Given how fast pathogens evolve and how serious drug resistance is becoming, could our growing ability to understand and maybe even deliberately shape our own human microbiota become our next great weapon in this ongoing arms race against infection?
Wow. Using our internal ecosystem as a defense shield.
Maybe. Or perhaps using specific beneficial microbes or their products as therapies. But that also opens up a whole new set of questions, doesn't it? Ethical considerations about manipulating our internal microbial world. What does it mean to be us if we start editing our microbiome?
That is definitely something to ponder. A fascinating and maybe slightly unnerving future possibility.
Indeed.
Well, thank you for joining us on this truly deep dive into the microscopic world that shapes us in so many ways. We hope you found it as fascinating as we did. Stay curious, everyone, and keep asking those big questions.